Okay, I'd like to get started. I think we're all here, and good morning for most of you. I'd like to welcome you to the Frontage webinar on biosimilars. My name is Matt Harrington from Frontage Marketing. It does have my boss's name, Ajay Sharma, as the host, but I'll be sitting in today. Our topic here today is an important one for us because we provide biologics development services to pharmaceutical companies that are investigating biosimilars. Frontage is a full-service contract research organization that moves large and small molecules from discovery through NDA. Thank you again for attending today. We'll follow up with you to discuss your individual project needs. Okay, I'd like to introduce Frank Diana first. He's our lead presenter. Frank is VP of Pharmaceutical Development at Endo Pharmaceuticals. He's also a professor in the Temple University School of Pharmacy, where he teaches a master's level class on biologics and biosimilars. After Frank's presentation, we'll hear from Ron Connolly from Frontage, and he will be discussing the biologics capabilities of Frontage and how we can help to move your compound forward. So I guess we'll get started. There's their titles, etc. So Frank, if you're able, go ahead and take it away.
Okay. Thanks, Matt, and good morning to everybody, wherever you are. Hopefully everybody hears me well. If not, let me know. So our topic today is biosimilars, and of course, as I'm sure everybody's aware, the reason that's of such interest at this time is because the government issued legislation in 2009-2010, which included discussion around biosimilars, and the FDA has been working on that since 2010, and in early 2012, they have issued three draft guidance documents that have been reviewed and are on their website in case you want to see them. And we'll go over all of that as we go along. So why is this such a big deal? Why don't we just get approval for biosimilars similar to generics? Well, first of all, a biologic is a lot more complicated and complex than a small molecule, and the generic approach is typical for small molecules but would not be typical for a biosimilar. So let's just start here right in the front of us. What is a biosimilar? Biologic product for which marketing approval will be granted based on less than a full safety and efficacy package. So in other words, you'd be able to utilize information that has been generated by a reference compound for a product that's already been approved in the United States by the FDA. It certainly would have to have physical and structural similarity to the already approved product, and we'll go into that in great detail as we go along. You would, as I said, rely on the some safety and efficacy of the approved product, with some exceptions, of course. And it's not a generic, as I mentioned, since cannot be characterized well enough to establish pharmaceutical equivalents and bioequivalents to reference drug. So you really cannot take the generic approach for a biosimilar. So just to make sure everybody's clear on the differences between a small molecule and a biosimilar and the path for approval, a small molecule drugs are approved through submission of an NDA using Section 505 of the FD&C Act, whereas biologics are approved through submission of a BLA, a biologic license application, through Section 351 of the Public Health Service Act. The FD&C Act has abbreviated approval pathway, the generic pathway, 505J, as everybody is aware of, and certainly if you're aware of the generic area, this is something you're very familiar with. However, the PHSA Act does not have that pathway, and that's why the legislation and the subsequent regulations have come out. So that is the future pathway for biosimilars. So again, what is this complexity around biotech drugs? Well, they have much higher molecular weights. Take the example of ibuprofen, our product that's used quite often, our NSAID product that's used quite often. That has a molecular weight of just slightly more than 200, whereas something like erythropoietin, has a molecular weight of over 30,000. And so you can see it's a heck of a lot more complicated with a lot of amino acids and also some carbohydrate chains, which is fairly typical for biotech products. So they have much more complicated structures, often complex with other biomolecules such as sugars and proteins and like the carbohydrates I mentioned for erythropoietin. They're more difficult to reproduce consistently due to more costly and complex production processes. So instead of using the organic synthesis that we would use in the case of small molecules, these are typically done through cell cultures and the like. And so they're a heck of a lot more complex in terms of manufacturing. There's a wide spectrum of molecular entities that fit into this category: polynucleotides, non-glycosylated peptides, complex proteins, antibodies, viruses, and so on. So there's a number of products that are considered biotech in nature. And this is just a simple diagram of sort of just showing the complexity of a diagram of the biologic product. So again, if we look at it, we see aspirin with very small molecular weight, very uncomplicated, and then as we proceed from HGH to IgG antibody, we increase the molecular weight and the complexity of the biologic quite a bit. And it's, an analogy would be to modes of transportation and how they are more complex as they are larger. So if we now, understanding a little bit of background on the biologics and biosimilars and why they're important, before I go into the Act, let's just even understand, so why is this such a big issue? Well, biologic products are expensive, as we all know. They're also some of our biggest products on the market today. Companies are selling large volumes of these products, and they're fairly costly. So the idea around the legislation is to give a pathway to other companies to come up with biosimilar products, and obviously then perhaps the cost may go down. And you'll see a lot of competition out there and a lot of different companies getting into this business. And so the approach from the legislation and FDA is to now give some guidance and control to that aspect. So in March of 2010, the Patient Protection and Affordable Health Care Act was signed into law, and it contains the Biologics Price Competition and Innovation Act of 2009, or what we're calling the BPCI Act, which amends the PHSA to create an abbreviated approval pathway for biologic products shown to be biosimilar to or interchangeable with an FDA-licensed reference biologic product, and that the pathway now will be PHS 351K for the biosimilar product. And so there's a lot of important words in there, and we're going to cover those. What is the difference between a biosimilar and an interchangeable product? It needs to be, you'll notice the words FDA-licensed reference biologic product, so it's something that the reference has been approved by FDA. Under the Act, a protein, except any chemically synthesized polypeptide, will be regulated as a biological product. And the guidance includes proposed definitions on proteins versus a synthesized polypeptide that we'll get into in a little bit. The Act also includes a 12-year period of exclusivity from the date of first licensure, that would be for the reference product. And then it also describes that the FDA was to come up with a user fee program for the fiscal years 2013 to 2017. And the fiscal year 2013 for the government starts in October of this year, so the FDA has been working on this area also, the user fee area. So the 351K application will include information demonstrating that the biologic product is biosimilar to a reference product based upon data derived from. And so then it goes into this stepwise approach, and the guidance documents also cover this and get into a lot more detail, but the Act itself talks about these things. So it describes the analytical studies demonstrating that the biologic product is highly similar to the reference product, and it does allow minor differences in clinically inactive components such as excipients and things to that nature. And on any of these, as you go along and we talk about the Act and the guidances, if any of these are, you think of questions about them, don't hesitate to pose them here, but also there is a Q&A as part of the guidance documents that the FDA has issued, and they have a lot of answers to specific questions that you might have around some of this information and how you would show biosimilarity and or interchangeability. So anyway, so analytical studies would certainly be necessary. And sorry, it looks like we jumped ahead here. And then we would move on to animal studies from there, including the assessment of toxicity. And again, we'll get into that in more detail. And then finally, a clinical study or studies, including the assessment of immunogenicity, pharmacokinetics, and pharmacodynamics. And in all of those cases, they would need to be sufficient to demonstrate safety, purity, and potency in one or more appropriate conditions of use for which the reference product is licensed. So this is your path for a biosimilar and an interchangeable. So first, you need to show it's biosimilar, and you're gonna show through analytical, animal, and clinical studies as detailed here. The application would also need to get into the fact that your product, your biosimilar, your proposed biosimilar would utilize the same mechanism of action for the proposed condition of use. Obviously only to the extent that it's known for the reference product. The conditions of use proposed in labeling have been previously approved for the reference product, so you're not asking for something new in your proposed biosimilar. It has the same route of administration, such as an injectable product, dosage form, and strength as the reference product, and strength is discussed in detail. All of this, those three, route of administration, dosage form, and strength, are discussed in detail in the quality considerations guidance document that the FDA has put out, and also there's some questions and answers covered in that guidance document. And of course, the manufacturing facility needs to meet the standards to assure purity, safety, and potency, so the typical GMP and such requirements in the manufacturing area, since the FDA will come in and do an inspection for a proposed biosimilar, as you might expect. So how similar does a biosimilar product need to be to the reference product? The subject product would be biosimilar if it is highly similar to the reference product, notwithstanding minor differences, as I mentioned before, clinically inactive components, and there are no clinically meaningful differences between the biosimilar product and the reference product in terms of safety, purity, and potency of the product. And this is going to be those set of studies that I had on the previous slide that you're going to do to get to that, and that's going to help you with biosimilar and, on the next slide, interchangeability. So interchangeability would be the next step up from biosimilar, and in this case, the subject product or the proposed biosimilar would be interchangeable with the reference product if it can be expected to produce the same clinical result as the reference product, and for a biological product that is administered more than once to an individual, the risk in terms of safety or diminished efficacy of alternating or switching between use of the biologic product and the reference product is not greater than the risk of using the reference product without such alternation or switch. So you can see the difference between biosimilar and interchangeability. So biosimilar, we show that it's acceptable analytically from an animal standpoint, and from a clinical standpoint, interchangeability, we take it up a notch and we say, well, you know what, we have to show that it's going to behave the same in patients. And certainly if it's going to be used more than once, it's got to be shown that if you alternate or switch, you're not going to see any differences with the reference product. So a biosimilar may get marketing approval without interchangeability with the reference product, and that's an important point. So you could have an interchangeable biosimilar or just a biosimilar, and there are some consequences of that. So again, if as I mentioned, the application for a biological product would be submitted under 351 of PHS Act rather than the 505 of the FD&C Act, and what the legislation has allowed is some what they call transition provisions, because there have been some classes of drugs, biologicals, that have been approved already under Section 505 of the FD&C Act, and we'll go over some of those later on. But anyway, the legislation says that they're going to provide this transition provision or an exception to this rule for anything that's been approved in a product class before March 23rd, 2010, which was when the legislation was signed into law. And they're going to give basically a 10-year period for that to sunset. So an application in these product classes may be submitted under 505 until March 23rd, 2012, unless there is another product licensed under Section 351 that can serve as the reference product if the application were submitted under 351K. So hopefully that's clear to everybody. Then they get into exclusivity, and again, keep in mind we're still talking about the original legislation. A lot of this, of course, is repeated in the guidance documents, but this is the original legislation that we're talking about here. And for exclusivity, the first interchangeable product, and notice that we're talking about interchangeable here, not biosimilar. Okay, so it's a product that's shown to be biosimilar and interchangeable. The first biologic product to be licensed as interchangeable is granted a period of exclusivity, and that's described very nicely in the legislation, and it's somewhat complicated and starts with a year and goes on, and it depends on what happens in the legal system and all that. So you'll have to read that closely, but there is a lot of information in there. During the exclusivity period, a subsequent biologic product relying on the same reference product cannot be licensed as interchangeable. So very similar to the way generics are treated. So if for a generic product that's the first to file and granted exclusivity, another generic cannot be approved until 6 months after the the original generic product. This is very similar with an interchangeable biosimilar product. Exclusivity dating is based on date of approval, date of first commercial marketing, and patent litigation milestones. So there's, again, the legislation goes into some detail around this. And again, there's some further detail in the question and answer guidance. With regards to exclusivity around the reference product. So first of all, a reference product is basically granted 12 years of exclusivity. So a 351K application may not be approved until 12 years after the date of first licensure of the reference product. And taken together with that, you cannot submit an application for a proposed biosimilar. You cannot submit an application for that until 4 years after the date of first licensure of the reference product. So that, in essence, it gives a time frame of 8 years between the 4 years and 12 years to file and try to get approval of a proposed biosimilar. Now, this does not apply to a supplement for the reference product. So subsequent application filed by the same sponsor or manufacturer of the reference product for a change that results in a new indication, route of administration, dosing schedule, dosage form, delivery system, delivery device, or strength. So it does not apply to that. Modification to the structure of the biological that does not result in a change in safety, purity, or potency. So the way you can read into that is that if you do modify the structure and it does change the safety, purity, or potency, that's likely, if it gets approved, to be approved as a new biologic and would again get the 12-year exclusivity. Okay, so we're going to move from the legislation now to the guidances. And so the legislation was from March of 2010. In between there, the FDA held some public meetings, and certainly many companies and organizations were able to comment on the legislation and on FDA's thoughts and how they were going to go forward. And from that, they were able to put forward a guidance document based on a lot of that feedback from industry and from different organizations across the spectrum. Anyway, they issued the 3 guidances. One is called Scientific Considerations in Demonstrating Biosimilarity to a Reference Product. And this is taken right off the FDA's website. This is basically exactly what it says. Draft guidance is intended to assist companies in demonstrating that a proposed therapeutic protein product is biosimilar to a reference product. And we'll go into some more detail on that, and it goes into the application called the 351K. And then the quality considerations in demonstrating biosimilarity to a reference protein product, and again, it goes into the details now, but more about CMC and analytical factors, whereas the top one was more around clinical and safety considerations. And then the last one is the one I've mentioned a few times, which is the question and answers regarding implementation. And again, it provides suggested answers, proposed answers to a variety of potential questions. And so that's what that goes into. So if we start with the scientific considerations draft guidance, and FDA, and basically FDA, gives a scope, and then they get into what is going to be their approach to biosimilarity. How are they going to evaluate your proposed biosimilar versus the reference product? So the document indicates that FDA intends to use what they call the totality of the evidence, and that's a very important statement in the document that you'll see, in its evaluation of the sponsor's demonstration of biosimilarity versus a previously FDA-approved reference product, and we'll get into that in a little more detail on the next slide. It recommends a stepwise approach to evidence development, ensuring that those elements necessary to address residual uncertainty are included. And basically what they're saying here is, for a proposed biosimilar, they're not expecting a application like it was a new biologic. You don't have to start from scratch. You are allowed to reference the reference product and utilize information from that. However, you're going to need to take care of the analytical, the animal, and the clinical studies that were described earlier to show that you have a product that is biosimilar. It gets into describing expectations for structure analysis, functional assays, and then moves on to animal studies and talks about the variety of things around animal studies that you might do. So it gets into some detail there. So you might, if you're interested in that area, that's where you'd go to look for animal studies and what variety of toxicology studies are recommended. And then it gets into a fair amount of detail on clinical study information, and it sort of steps you through these different clinical aspects, human pharmacology, so PK, and pharmacodynamic studies that we mentioned earlier, clinical immunogenicity assessment, which we'll talk about again in a little bit, but that's obviously extremely important from a safety standpoint, and then clinical safety and effectiveness data, talks a little bit about design issues and how you would design those studies in terms of inferiority and superiority and things like that. It talks some about extrapolation across indications and if you can do that and when you can do that and what's the justification for doing that. And the question and answers document also goes into some detail around that. And finally, it discusses post-marketing safety considerations and what's the expectation for monitoring adverse events once a biosimilar is approved. So as I mentioned, this is a slide that gets into more detail on the totality of the evidence, and I wanted to mention that we took this slide directly from an FDA presentation from February 15th, 2012, and you can see that presentation on the FDA's website, but this slide comes directly from that presentation. And basically just shows the foundation is clearly the structural and functional characterization, a lot of the analytical work that will go into showing comparability between the proposed biosimilar and the reference product, and then continues with human PK and PD studies, on to clinical knowledge, clinical immunogenicity, animal studies, and at the top, clinical studies. So from highly similar to biosimilar. And as it says here on the side, no one-size-fits-all assessment, and that certainly the sponsor and the FDA sponsor will do what they believe is correct to show that what they believe is a biosimilar product, and then the FDA will evaluate the applicant's integration of the various types of information to provide advice and scope and extent of development plan, and ultimately an overall assessment that a biologic product is or is not biosimilar to an approved reference product. So that's what that slide gets into. Totality of evidence is the key to this particular guidance. So what are some of the key scientific issues that the industry knows is out there and that the guidance is trying to get into? Well, as we've talked about, biosimilars are made in complex biologic systems, and thus it's impossible to manufacture an exact copy of the reference biopharmaceutical, and thus we're going to show they're biosimilar. Changes in the structure can occur during production process, leading to differences in efficacy, immunogenicity, and safety. Proving similarity with the reference product is, as we know, a major challenge, but is certainly doable. Assays for biologics are less sensitive and precise than tests for small molecule drugs, so you're going to see some variability in the testing. And how do we take that into account when we're trying to show a reference product and a biosimilar are basically biosimilar, a proposed biosimilar? Non-clinical and clinical data are needed to determine safety and efficacy, and need for post-marketing surveillance and risk management. So all of those are features discussed in this, and certainly the industry is well aware of them. So another area of concern is this immunogenicity and patient safety. As we very much know, biologics and biosimilars have potential to provoke an immune reaction, and often the preclinical assays are not sufficiently predictive of the immunogenicity in humans. So animal studies typically are not going to be that predictive of what could happen in humans. And so, it's a concern, and there are several factors that can influence potential for immunogenicity, including impurities that could be in the potential, or the proposed biosimilar to the reference product, so there could be differences in the impurities. Structural modifications introduced during manufacturing and storage could play a role in immunogenicity, and the route of administration, and of course the fact that we have a variety of patients and how they react could also play a big role. So you'll notice as this legislation is debated and these guidances are debated that immunogenicity will play a large role in that debate, and the fact that patient safety, of course, is of utmost importance. So then we're going to turn to the quality considerations draft guidance. So that's the second guidance, and basically it again goes into gives you the whole preliminary discussion again on the BP, on the act itself and the legislation, and then it gets into describing, well, you need to have a complete and thorough CMC section, which I don't think surprises anybody, and then it gets into focusing on the analytical studies that may be relevant to assessing the similarity between two protein products, so the proposed versus the reference. And it also, as I mentioned earlier, gives a definition for protein via chemically synthesized polypeptide definition. It's somewhat controversial. It's basically based on size, 40 amino acids versus 100 amino acids. And you could take a look at that, but remember these documents are draft. This particular definition was not in the Act itself. This is a guidance, a draft guidance that the FDA has proposed, so we'll see where that ends up eventually. And then it gets into a number of factors for consideration. Each one is discussed in some detail. The expression system, the manufacturing process itself, the fact that the importance of the physical chemical properties and the functional activities and how you measure those and what types of tests that you do. Impurities gets into doing, of course, drug product stability and how you would go about doing that, just that you would need to do that, and obviously that's covered by many ICH and FDA documents. Identification of lots used in the various analyses for biosimilarity determination. So that's another important thing. How do you show that a reference product, or your proposed biosimilar is indeed similar, highly similar to the reference product? Well, you're going to have to show that through a variety of lots, and that's the expectation there, and that's why it talks about identification of lots. Some of the things that the quality guidance is trying to cover, and just some of the background, I guess I'd call it. That's sort of the way I would describe this slide. It says that, what was sort of the background that led up to a lot of this, and why are the guidances the way they're written, and what is the issues that we face around the quality side? Well, we know that empirically there's evidence that shows that process and formulation changes can have a significant impact on safety and efficacy of the product. There are many examples for reference drugs that highlight these issues, and can occur due to changes to the biologics. There is a ICH guidance that talks about this. It's called ICH Q5E. I included it as a reference document at the end that describes what you would do for manufacturing changes. So this is just sort of a background or a foreshadowing of some of the difficulties that a proposed biosimilar brings to you. These are, you might say, are things where we might even have a lot more knowledge about a product, because this would be the same company making changes, and so they could have a great deal of knowledge about a product, whereas a manufacturer for a proposed biosimilar may have limited knowledge at first as they develop their biosimilar. And even with all of that knowledge that the reference product manufacturers may have had, they still had issues such as, so here's an example, a product called Raptiva, which the idea was just they were going to scale up the production, and as it turned out, when they scaled up, the changes affected molecular structure and the pharmacokinetic profile. You ended up at higher serum concentrations, and in the end, a phase 3 clinical trial had to be done to confirm safety and efficacy, just really for scaling up the product at the same company. Second example would be Eprex, which is a glycoprotein therapy for anemia, and there was a production process change. This was almost 15 years ago that this occurred, so in the late 1990s, early 2000s, and when that change went into effect, it included reformulation and a change in the container closure, and at that time there was seen a higher incidence of immune reaction leading to pure red cell aplasia, a very dangerous issue. And at that time, the company went into a full investigation mode, and it was found that most cases were subcutaneous administration in patients with chronic kidney disease. It just highlighted the importance of several things, including the storage and handling, so the cold chain, since a lot of these products are injectables and biologics, a lot of them are stored at refrigerated temperatures and the like. In the end, the investigation, I'm not sure there was ever a complete conclusion, but some of the ideas were organic compounds leached from uncoated rubber stoppers and pre-filled syringes containing polysorbate 80 as the most probable cause of the increased immunogenicity. So again, the concern in the industry and at FDA around changes to products, and that also you could see the same in with regards to biosimilars. A third example is a product called Mysozyme. This is for a treatment of Pompe disease. This is a more recent issue. There was a scale-up at a new facility, and in April 2008, the FDA rejected it. The scale-up, the original approval for the product had been granted in 2006. It's, again, a fairly large glycoprotein, as you can see. The original scale was a lot smaller. The proposed scale was to be increased quite a bit, and it had already been approved in many countries outside the U.S. FDA felt that the carbohydrate chain is different between the two products, and therefore the BLA required for new sites scaled-up process, including clinical data. And this has gone on and on for the last several years, and I believe recently there was an approval in this area for a scaled-up version, although it wasn't exactly as originally described. So again, just sort of a foreshadowing background to the complications with biosimilar products. Again, FDA has provided some biosimilar statistics, as I call them, and you can see there what some of the things that have been going on: 35 pre-IND meeting requests for proposed biosimilar products to 11 reference products, 21 pre-IND sponsor meetings held to date, development programs, including prospective development, global programs, retrospective development programs seeking licensure in U.S. for similar biologic products licensed outside the U.S., 9 INDs received. So these are just a variety of things the FDA has been getting requests for and dealing with over the last few years. So again, just a little more background and where a lot of this is coming from. This is just an interesting product called Omnitrope, which was, you might argue, was the first biosimilar approved in the U.S. However, it ended up getting approved as a new biological entity, as it could not be scientifically proven to be equivalent to the reference product, genotropin. And if you look at that, and so it ended up getting approved under the 505(b)(2) pathway that we talked about earlier. And similar other products, Valtropin, which was biosimilar to Umatrope, and then a variety of other products, all approved under the 505(b)(2). And if I just go into Omnitrope as an example of all of these, just to give you just a little bit of background in this area. So Omnitrope is an injectable product that's a recombinant human growth hormone. It's a single polypeptide chain, so it's fairly simple. You could see that there was an application, and the approval mechanism turned out to be a 505(b)(2) for the adult indication. And so the sponsor was utilizing some information from the reference product. And there were those indications as written there. They're not particularly important to this discussion. And just running through some of the timeline for Omnitrope and the complications as it went through the review process, you could see it was filed in 2003, and there were some citizens petitions, CPs filed, that were requesting FDA to refuse to approve any application that was using information contained in another approved application for biologics. And then we went on, and FDA completed their review but was unable to make a final decision. And if you go look at the FDA website and you look up this drug, you'll see the review, and it's interesting. You'll see that it talks about how the reviewer concluded what they concluded in 2004, but no final decision was reached. Then there was a lawsuit, and then finally a federal judge ruled that there must be a decision, and then at that point the FDA approved Omnitrope biosimilar, or according to the 505(b)(2), and then shortly before that it had already been approved in EU as what's considered the first biosimilar in Europe. So again, this is just some background to what are some of the complications around the legislation. The reason why Omnitrope was seen as an example of something that was fairly simple to approve by this route is because it's well characterized. There's no glycosylation, so it's not a complex molecule. Its structure is easily determined, or can be determined with current physical chemical tests. It has clinically relevant bioassays and qualified biomarkers. It's a long and well-documented history, and its mechanism of action is known. And so that was a lot of the reasons provided by FDA for approval, ultimately, of the product in 2006. And again, that's just given as a background to this whole issue around biosimilars. It was not considered a therapeutically equivalent, so it was not AB rated, so it was not a generic. And subsequent, and even before this, several other follow-on protein products were approved under the 505(b)(2) pathway. And that's part of the reason why there's those transition principles that the FDA and the legislation provides. So just briefly going into user fees. So the legislation indicated that user fees should be set up, and there should be an initial 5-year period for that, 2013 to 17, as I mentioned. The difference with these user fees and the typical user fees we're used to in the industry is these would be provided at the early stages of development. So when you filed an IND, you would be paying a user fee, and then subsequent each year you'd be paying a user fee. And of course, they're not as large as the user fees you pay for a current NDA. They're sort of split over several years. It should ensure adequate resources for review so that critical resources are not redirected from review of current biological applications. So that was part of the legislation. So that's the whole reason for, okay, the user fee program for biosimilars. So as I said, it was paid upon submission of the IND, and there's also other fees that are also included. And in the end, you would pay a similar fee to what we would pay for a small molecule, but it would be, you would obviously subtract the previously paid amounts. It then goes into performance goals. We won't go into those here, but they are on the FDA website. You can look at those, and you know, the FDA says they'll review biosimilar applications within 10 months for 70%, and so on and so forth, and it sort of scales up till you get to 2017. It's more like what you see for small molecules. And then just a couple of additional topics that are out there and being discussed with no firm guidance or anything, but obviously the naming of biosimilars and interchangeable products, how will that be handled? If we're going to have safety, how would we keep track of safety of an interchangeable product if it doesn't have a different name? And then notification to the reference product, how would that be handled from a legal standpoint? Would it be similar to generics? Again, a lot of that's covered in the legislation, but there are still some open questions around that.
Well, great. Thank you, Frank, so much for the informative discussion this afternoon. And this is Ron Connolly. I'm Senior Vice President for Frontage. I just wanted to take 2 minutes to review some of the services that Frontage can provide in development of biosimilars, but I also wanted to give everyone an opportunity to ask a question. If you'd like to ask a question with regard to the presentation, please feel free to use the Q&A section of the webinar here. Otherwise, you can send us an email with your questions and we'll try to answer that. We will also have the presentation available for you after the discussion today. So briefly, Frontage offers several key areas of service to help companies in development of biologics and biosimilar products. The first key area is in our biologics methods and development and testing services. We also are helping companies with reverse engineering the current products that are on the market and developing multidisciplinary comparability assessments using various techniques, both physical, chemical, and some other techniques to help to reverse engineer the products. And then we offer regulatory support as well. We have personnel that have experience with biological products that can help with developing the comparability rationale, that can help with pharm tox and clinical pharmacology rationale when you're speaking with the FDA, and I'll dive into that a little bit in a second. We also are offering some clinical support services as may be suited for certain types of products. So moving into the biological services area, we are offering assay development and method validation, and that includes, developing antibodies that may be suitable for a biologic that you are developing. We are doing, at this time, both clinical and preclinical sample testing that is GLP compliant and CLIA certified, and our facilities are FDA inspected and are working on a number of biological programs at this time. We also are offering within the realm of the clinical sample testing, we have the PD, PK, and TK sample analysis. So we are developing various types of pharmacokinetic assays in support of the animal studies and product development studies. We are also developing immunogenicity tests, which may include these two different types of platforms using anti-drug antibodies or neutralizing-based antibodies. We are also doing cell-based assays and assays for biomarkers, which are really important for evaluating the pharmacodynamic properties of the biologic that you are studying. And finally, we are also offering mass spectrometry assay development and testing. We actually have two programs ongoing where we've successfully developed mass spec methods for use in biological assay testing for pharmacokinetic assessments. And there's some advantages for these kinds of assay platforms over the traditional ELISA-based platforms considering the fact that you may have a reduced risk in cross-reactivity, in platform variability, and also you can improve some specificity in the protein or the antibody that you're evaluating. Our biologic services areas encompass the 3 key areas, and in detail, the bioanalysis supports pharmacokinetics and toxicokinetics while you're doing those evaluations of the biologic in human and animal subjects. In support of the PK and TK data, we also are working on immunogenicity assessments, and that's where you're evaluating the immunoresponse of the drug. And it's a complicated area because it requires one to develop the cut points of a positive response and a negative response in immunogenicity inside the subjects being evaluated. So there's some complicated method development that's involved in determining what that cut point is for a positive response or negative response. And then we are also offering the biomarker and pharmacodynamic analysis, and that's helping you to evaluate, what is the clinical response of the drug in a subject. Some of the, I also put up this slide because developing a biological assay is quite complex. It's much more complex, as Frank pointed out earlier. The size of the molecules are very large, and so there are a number of considerations that have a tremendous impact on the variability in the assay. So these are just a few of the examples of the thought process that goes into developing a biological assay. And Frontage has hands-on experience dealing with all of these issues and making sure that you have a robust method, time in and time out, for each time point and sample that's generated during the clinical development and preclinical development of a biologic. Another area in which we're offering support is in our regulatory services area. We have experienced individuals that have worked on biologics and small molecule programs, and we are helping companies to file INDs and NDA filings, and of course the biosimilar area will be working in that platform to file applications as well. We not only file the applications, but we also work together with our clients to really develop a rationale and approach to how to go to the FDA and negotiate with them on the comparability of your biosimilar to the reference product. And this is very important because if you go into the FDA without a rationale ahead of time, it allows the FDA to be able to make a lot of commentary on your approach to how you're doing your comparability and how you're doing your clinical design. So this takes a lot of thought and a lot of thought leadership to work through this area. We are helping companies to develop comparability protocols to make sure that you have a good rationale on what kind of physicochemical tests that will be taken, the animal testing, and the clinical program. We're helping with the clinical pharmacology strategy and making sure that the sizes of the studies are appropriate to get the statistical data that you need to show the FDA that you have a biosimilar, and helping the companies with connecting them with key opinion leaders in these certain therapeutic areas that the biologics are used in. Overall, Frontage has 300 researchers in the U.S. and in China, and we're supporting companies beyond just biosimilars, helping with preclinical and DMPK studies, the drug development aspects of formulation, analytical, and active ingredient manufacturing, and we are supplying clinical trial materials to companies that have programs in clinical development. We also support companies with bioassays, biological testing, and we have clinics both here in the U.S. and in China to introduce product applications to both of those markets for approval. And finally, we work together with companies with regulatory affairs and GMP consulting. You can find out more about Frontage by visiting our website, www.frontagelab.com. And I'm going to hand it back over to Matt. Thank you for your attention today.
Thanks, Ron. Good job. I also noticed there is one question that maybe Frank can address from Amy Belton. At what point during biosimilar development, analytical, animal, and clinical studies should the manufacturer request a meeting with the FDA? Frank, any thoughts on that one?
So yes, actually the question and answers guidelines goes into that quite a bit. I believe it suggests when you have enough information, first of all, you have a plan as to how you're going to approach your biosimilar, showing that your product is biosimilar, and you already have some comparable data between the biosimilar and the reference product. I believe it goes into then that's a good time to request a meeting with the FDA. But there is, like I said, you can see an answer to that I'm looking for right now. But anyway, you'll see there's a question related to that exact question, and that's the answer that the FDA is proposing at this point.
Okay, thanks, Frank. And any other questions about? Amy had mentioned what's the typical bioservices package should be addressed separately and individually, and so we'll follow up with you directly, Amy, on that one, because it is a long answer and specific to your needs. So also there's a question about the slides. Yeah, the slides will be shared with all participants, and you can actually, in the screen now, if you go up to the File pull-down menu, the drop-down menu, and just save documents, you can save it to your hard drive that way. So go ahead and do that now if you'd like, or we will have the slides available on our website, and we'll send them out individually to those participants, okay? At this point, are there any other questions from participants or thoughts, contributions from anyone?
Matt, this is Frank. I see a couple of questions on the website.
Okay.
And one of the questions is, has FDA come up with specific interchangeability requirements, or what determines interchangeability? So no, that's their totality of evidence approach. And basically, first you have to show biosimilarity, and then then the question will be, will it be interchangeable? And that gets into, you need to show no effect in switching products, as was described in the definition of interchangeability. So that would probably require some additional studies to get to that. So I would assume that would be somewhat the answer to that, and that's going to be a very important piece, is getting a biosimilar to be interchangeable. And then what would be the biosimilar cost? Okay, well, that's a good question. Depends on who you listen to. I see a variety of things that run from anywhere from, if a biosimilar is approved, of course that's going to be specifically a company decision, and it will depend on how many products eventually are approved and things like that. But I've seen numbers in the area of 50 to 90% of the cost of the reference drug, depending on which company you're talking about, and these are all companies that don't have a biosimilar at this point, but that's what they're thinking anyway.
Yeah, and Frank, I also wanted to add a point that each product has very unique characteristics. So this allows the FDA to customize what they mean by interchangeability when it comes to the comparability of your product to the reference product. So we've seen in some cases where there's very fine detail in determining the properties of that biologic matter from structural elucidation to particle size, to even how the material transfers through a membrane, and what kind of molecular weight distribution is in the product that you're offering. So there's a lot of different considerations given in that regard.
All right, thank you again, everybody, for attending today. Please, again, visit our website for more information about Frontage. Also, keep an eye out for any future webinar announcements. We'll keep you on our list. And Frank or Ron, anything else you wanted to add before closing?
No, not me. Thanks for your attention.
Yeah, thank you so much, Frank. I know you took a lot of time to prepare this presentation for everyone, and it was a very good synopsis of what's happening right now.
All right. Take care, everyone. Thanks again. Bye-bye.
Thank you. Bye.